Number 1. The killer that erased an entire hemisphere. In the autumn of 1520, a Spanish ship anchored off the coast of Mexico. On board was an enslaved African man whose name has not survived. He had been recruited into the army of Pánfilo de Narvaez, sent by the governor of Cuba to arrest Hernán Cortés. The man was not a soldier. He was carrying something the Spanish themselves did not yet understand. He was carrying smallpox. Variola virus is an orthopox virus, a large double-stranded DNA virus in the family Poxviridae. The lethal form, variola major, kills roughly 30% of those it infects. Survivors are scarred for life. Phylogenetic studies place its emergence as a human-specific pathogen at 3,000 to 4,000 years ago, derived from a rodent-borne ancestor. The earliest possible evidence is the mummy of Pharaoh Rameses V, who died around 1145 before the Common Era, and whose face still bears pustular lesions consistent with smallpox infection. By 1520, smallpox had been killing Europeans, Africans, and Asians for thousands of years. Most adults in the Old World had survived it as children. They carried lifelong immunity. The people of the Americas had never encountered it. The first outbreak in the New World struck Hispaniola in 1507 or 1518, devastating the Taino population. By the time the virus reached Mexico in 1520, Cortés was besieging Tenochtitlan, the Aztec capital. The Aztec emperor Cuitlahuac, who had succeeded Moctezuma II, contracted smallpox and died within weeks. The siege ended as much from disease as from steel. Modern estimates suggest 40% of central Mexico's population died in that single outbreak, 5 to 8 million people. Smallpox reached the Inca Empire in 1524, killing the emperor Huayna Capac and triggering a civil war between his sons Atahualpa and Huáscar. By the time Pizarro arrived in 1532, the empire was already broken. Pizarro did not conquer the Inca. He walked into the wreckage smallpox left behind. For the next 300 years, the same pattern repeated across the hemisphere. New England, 1633, the Great Plains, 1780, and again in 1837. The Pacific Northwest, 1862. The total indigenous American population declined from contact-era diseases, primarily smallpox, with measles, influenza, and typhus alongside, is estimated at 80 to 95%. From roughly 60 million people before contact to about 5 to 6 million by 1700, the largest demographic catastrophe in human history. In 1798, the English physician Edward Jenner published his inquiry into the causes and effects of the variolae vaccinae, demonstrating that exposure to cowpox produced immunity to smallpox. He coined the word vaccine from the Latin vacca, meaning cow. It would take another 170 years to turn his observation into eradication. The World Health Organization launched its intensified smallpox eradication program in 1967 under Donald Henderson. The last natural variola major case was Rahima Banu in Bangladesh, October 1975. The last natural variola minor case was Ali Mau Malin in Somalia, October 1977. Smallpox was declared eradicated by the World Health Organization on May 8, 1980. It is the only human disease ever eliminated. 2 declared stockpiles of live variola virus still exist at the Centers for Disease Control in Atlanta and at the Vector Institute in Koltsovo, Russia. They are the last of their kind, officially. Smallpox is the only human disease ever eradicated. The next virus on this list is the only retrovirus that has matched it for cultural impact. It crossed from a chimpanzee less than 120 years ago. Number 2. The retrovirus that crawled out of the jungle. Somewhere in southeastern Cameroon, around 1908, a hunter cut himself while butchering a chimpanzee. The chimpanzee was infected with simian immunodeficiency virus, or SIV. The hunter contracted what would eventually be called HIV. He almost certainly never knew. He may have lived another 20 or 30 years before the disease killed him, and during that time he passed it to others. HIV is a lentivirus in the family Retroviridae. Like all retroviruses, it carries its genome as RNA and uses an enzyme called reverse transcriptase to convert that RNA into DNA, which it then integrates permanently into the host cell's chromosomes. Once HIV is inside a cell, it cannot be removed. The geographic origin was traced by Beatrice Hahn's group at the University of Alabama at Birmingham in 2006. Through phylogenetic comparison of HIV 1 group M with simian immunodeficiency virus strains in chimpanzees, Hahn's team narrowed the source to chimpanzees of the subspecies Pan troglodytes in the upper Sangha and Bumba River basins of southeastern Cameroon. In 2008, Michael Worobey and colleagues published a molecular clock analysis in Nature dating the cross-species transmission to approximately 1908, with a confidence range from 1884 to 1924. The transmission mechanism is called the bushmeat hypothesis: cutaneous or mucous membrane exposure to infected ape blood and body fluids during hunting and butchering. It is not exotic. It is the same way humans have always interacted with the animals they kill for food. For the next 50 years, HIV circulated in Central Africa unrecognized. The earliest confirmed HIV-positive sample is known as ZR59, taken in 1959 from a man in Leopoldville, the city now called Kinshasa, in what was then the Belgian Congo. Worobey's reconstruction shows the epidemic radiating from Kinshasa beginning in the 1920s and 30s. Colonial-era medical injection campaigns, in which the same needles were reused across hundreds of patients, may have amplified early transmission. The virus reached Haiti in the 1960s, carried by Haitian professionals returning from work in the Congo. From Haiti, it reached the United States in the 1970s. On June 5, 1981, the United States Centers for Disease Control reported in the Morbidity and Mortality Weekly Report a cluster of 5 gay men in Los Angeles with pneumocystis pneumonia. It was the first recognized cluster of what would soon be called AIDS. The original name was GRID, gay-related immune deficiency. It was renamed in 1982 when heterosexual, hemophiliac, and pediatric cases made the original framing untenable. Luc Montagnier's group at the Pasteur Institute isolated HIV in 1983. Robert Gallo at the United States National Cancer Institute confirmed and characterized the virus the following year. The priority dispute was eventually settled with shared credit. The 2008 Nobel Prize in Medicine was awarded to Montagnier and Françoise Barré-Sinoussi. Cumulative deaths from HIV/AIDS are approximately 40.4 million as of 2023. Approximately 39 million people are currently living with HIV worldwide. Modern antiretroviral therapy can suppress viral load below detection limits, effectively eliminating sexual transmission risk under what is called the U equals U principle. Undetectable equals untransmittable. Pre-exposure prophylaxis, or PrEP, has been available since 2012. SIV variants infect more than 40 species of African primates, mostly without causing disease. Only the cross-species jump to humans, and to a lesser extent to certain macaques, produces immunodeficiency. The virus that has killed 40 million people is, in its natural hosts, harmless. HIV crossed from chimpanzees in 1908. The next virus crossed from mosquitoes hundreds of years earlier. It ended Napoleon's New World Empire before he ever set foot on it. Number 3, the mosquito-borne killer that defeated Napoleon. In 1801, Napoleon Bonaparte sent his brother-in-law Charles Leclerc to the Caribbean colony of Saint-Domingue with an army of 40 to 60,000 French soldiers. Their mission was to crush the slave revolt led by Toussaint Louverture, restore French control of the colony, and reassert France's grip on the New World. Within a year, more than half the army was dead. They had not died in battle. Yellow fever is caused by a flavivirus transmitted by Aedes aegypti mosquitoes. The virus likely originated in Central or East Africa and crossed the Atlantic in the 17th century via the slave trade. Aedes aegypti itself is also of African origin, transported to the Americas in the water barrels of slave ships. The first major American epidemic struck Barbados in 1647. The disease is cinematic in its cruelty. After an incubation of 3 to 6 days, the victim develops sudden high fever, chills, headache, and severe muscle pain. Within days, the liver begins to fail, producing the characteristic jaundice that turns the skin and eyes a sickly yellow. In severe cases, the gastric lining hemorrhages, producing the signature symptom that gave the disease its old name in Spanish, el vómito negro, the black vomit. Mortality among non-immune adults reaches 25 to 50%. Survivors gain lifelong immunity. When Leclerc's army landed in Saint-Domingue, almost none of them had ever encountered yellow fever. The local Creole population, both enslaved and free, had higher rates of immunity from prior exposure or partial inherited resistance. Within months, French troops were dying by the thousand. Leclerc himself died of yellow fever in November 1802. Napoleon dispatched a second wave of reinforcements. They died too. By the time the campaign collapsed in 1803, an estimated 50,000 French soldiers had been killed by the mosquito. Napoleon abandoned his plans for a new world empire. In April 1803, France sold the Louisiana Territory to the United States for $15 million. 828,000 square miles, doubling the size of the United States overnight. On January 1st, 1804, Haiti declared independence. It became the first black republic and the only successful slave revolt in modern history. A flavivirus delivered by a mosquito had rewritten the political map of two continents. The historian John McNeill, in his 2010 book Mosquito Empires, argued that yellow fever and malaria together created what he called a mosquito empire, a biological barrier that protected Caribbean Creole societies from European invasion for over a century. The same disease ecology shaped the geography of the slave trade in horrifying ways. West African populations had higher rates of partial immunity to yellow fever, which made them disproportionately valuable to slavers. The biology of a virus dictated which human beings were trafficked across the Atlantic. Yellow fever did not stay in the Caribbean. In 1793, an outbreak in Philadelphia killed 5,000 of the city's 50,000 inhabitants and forced the temporary relocation of the federal government. The Cuban physician Carlos Finley proposed the mosquito vector hypothesis in 1881, a claim widely dismissed at the time. It was confirmed by the Walter Reed Yellow Fever Commission in 1900 through human volunteer experiments. One member of Reed's team, Jesse Lazier, allowed himself to be bitten by an infected mosquito to test the hypothesis. He died. Max Thieler developed the 17D vaccine in 1937, earning the 1951 Nobel Prize in Medicine. Yellow fever remains endemic in tropical Africa and South America. Before recent vaccination efforts, the World Health Organization estimated 200,000 cases and 30,000 deaths annually. Yellow fever killed armies. The next virus killed children. It had been doing it since the pharaohs. Number 4. The paralyzer that stalked humanity for 3,000 years. In the Nye Carlsberg Glyptotek Museum in Copenhagen, there is an Egyptian funerary stele dated to roughly 1400 before the Common Era. It is called the Stele of Roma, sometimes Ruma's stele. It depicts a priest leaning on a staff, his right leg withered and atrophied to a fraction of the size of his left. It is the oldest depiction of a viral disease in human history. The disease is polio. Poliovirus is an RNA virus in the genus Enterovirus, family Picornaviridae. It exists in 3 serotypes, type 1, type 2, type 3. Transmission is fecal-oral. The virus replicates in the gut and is excreted in the stool. Most infections cause no symptoms or only mild illness. But in roughly 1 in 200 infections, the virus invades the central nervous system, destroying motor neurons in the spinal cord and producing the irreversible paralysis the world came to fear. Polio almost certainly afflicted humanity for thousands of years before it became a recognized epidemic disease. Possible polio cases have been identified in several Egyptian mummies, including the 12th dynasty pharaoh Siptah, who died around 1190 before the Common Era, and whose mummy shows a clubfoot consistent with polio sequelae. Michael Underwood, an English physician, gave the first clinical description of polio as a distinct disease in 1789. Then, in the late 19th and early 20th centuries, something changed. Major polio epidemics began to emerge, not because the virus was new, but because the world was. The link, paradoxically, was improved sanitation. In societies with poor hygiene, infants encountered poliovirus very early in life, while still protected by maternal antibodies. The infection caused mild illness and conferred lifetime immunity. As clean water and sewage systems eliminated infant exposure, the first encounter shifted into childhood and adulthood, when maternal antibodies were gone. The virus that humans had carried for millennia became, almost overnight, a paralyzer of children. The 1916 New York polio epidemic killed over 2,000 people, mostly children, and triggered widespread public panic. Franklin Delano Roosevelt contracted paralytic polio in 1921 at age 39. He lost the use of his legs for the rest of his life. He founded the Georgia Warm Springs Foundation in 1927, and what became known as the March of Dimes in 1938. The iron lung mechanical respirator, which kept paralyzed victims breathing, was invented in 1928 by Philip Drinker and Louis Agassiz Shaw at Harvard. The 1952 American polio epidemic was the worst in United States history. 57,628 cases, 3,145 deaths. 21,269 permanently paralyzed. 3 years later, on April 12, 1955, the inactivated polio vaccine developed by Jonas Salk at the University of Pittsburgh was declared safe and effective. By 1961, Albert Sabin had developed the live attenuated oral polio vaccine, which became the global eradication tool because it was easier to administer and produced herd immunity through community transmission. The Global Polio Eradication Initiative was launched in 1988 by the World Health Organization, UNICEF, Rotary International, and the United States Centers for Disease Control. Wild poliovirus type 2 was declared eradicated in 2015, type 3 in 2019. Only type 1 remains endemic in just 2 countries, Pakistan and Afghanistan. The polio vaccine race established the modern model of global public health infrastructure, mass clinical trials, vaccine development pipelines, and coordinated international immunization programs. It also created the modern complication of vaccine-derived poliovirus outbreaks, which occur when the attenuated oral vaccine reverts to virulence in under-vaccinated populations. Polio's killer was paralysis. The next virus's killer was the empire it broke and the religion it accelerated. Number 5. The pox that broke the Roman Empire. In 165 of the Common Era, the Roman legions were returning home. They had been campaigning in Mesopotamia under the co-emperor Lucius Verus in what historians now call the Parthian War. The war had gone well. The legions had taken Seleucia and burned the temple of Apollo at Babylon. They were marching back to Rome with their plunder. Something marched with them. The Antonine Plague struck the Roman Empire from 165 to roughly 180 of the Common Era, named after the reigning Antonine dynasty of Marcus Aurelius and Lucius Verus. The pandemic almost certainly began in the army camps of Mesopotamia. Returning soldiers carried the disease back to Rome, then outward across the empire's network of military roads, which functioned as the most efficient pathogen distribution system the ancient world had ever built. The physician Galen of Pergamon was practicing in Rome at the time. He treated victims and recorded clinical observations that have come down to us in his surviving works. He described pustular eruptions covering the body, high fever, and a black exanthemia lasting around 9 days. Most paleomedical scholars, including R.J. Littman and R.P. Duncan Jones, interpret Galen's descriptions as consistent with smallpox, the same variola virus that would, more than 1,000 years later, end the Aztec and Inca empires. A minority view holds that measles is also possible given the febrile and exanthematous nature. The death toll is estimated at 5 to 10 million people, roughly 7 to 10% of the entire population of the Roman Empire. The co-emperor Lucius Verus died in 169 of the Common Era, possibly of the plague. Marcus Aurelius died in 180. The historian Cassius Dio attributed his death to the recurrent plague, though some sources point to a separate illness. Either way, the rulers of the empire were not spared. The Roman army suffered catastrophic manpower losses. The frontier defenses against Germanic tribes weakened during the Marcomannic Wars. The empire was forced to recruit gladiators, slaves, and barbarian conscripts to refill the legions. Egyptian papery from the period document farmland abandonment, severe labor shortages, and a sharp decline in tax revenue. The economic damage rippled across the Mediterranean. The sociologist Rodney Stark advanced an influential thesis about a downstream consequence. The Antonine Plague accelerated the spread of Christianity, partly because Christian communities organized care for the sick when civic structures failed. Roman elites fled cities during outbreaks. Christians stayed and tended the dying, both their own and pagans. The result was both a higher survival rate among Christian communities and a powerful conversion narrative for the religion's growth. A second pandemic, the Plague of Cyprian, struck the empire from 249 to 262 of the Common Era. It was named after Cyprian of Carthage, a bishop who described its symptoms. Some historians interpret it as a recurrence of the Antonine pathogen. Others argue it was a different disease, possibly a hemorrhagic fever. It compounded the damage. Modern attempts to confirm the pathogen of the Antonine Plague through ancient DNA have been inconclusive. A 2020 study by Barbara Bray Manti and colleagues attempted to extract pathogen DNA from Roman plague-era burials but did not find conclusive evidence. By contrast, variola DNA has been successfully recovered from a 17th century Lithuanian child mummy by Duggan and colleagues in 2016 and from 11 Viking-era skeletons by Muleman and colleagues in 2020. The ancient DNA evidence confirms smallpox in early modern and medieval Europe. It just hasn't reached Rome yet. The Antonine Plague broke an empire. The next virus broke entire continents. It killed every child it touched. Number 6. The cattle plague that became humanity's great child killer. Measles is so fragile in epidemiological terms that it cannot exist without us. The virus needs a constant supply of unexposed people to infect. In small populations, measles burns through every available host and then dies out. The English statistician Maurice Bartlett calculated in 1957 that measles requires a continuous population of 250,000 to 500,000 people to maintain endemic transmission. Below that threshold, the virus cannot persist. He called this the critical community size. This means measles is younger than civilization. For most of human history, our populations were too small and too dispersed to support it. The first cities to cross Bartlett's threshold appeared in the Bronze Age, Mesopotamia, the Nile Valley, the Indus, and it was in those early cities, surrounded by domesticated cattle, that the ancestral measles virus made its jump. Measles virus is a member of the genus Morbillivirus, family Paramyxoviridae. Its closest known relative is rinderpest virus, the cattle plague, also a Morbillivirus. For decades, scientists assumed measles had emerged from rinderpest sometime in the medieval period, perhaps the 11th or 12th century of the Common Era. In 2020, a study published in Science by Ariane Dux and colleagues used a measles virus genome recovered from a 1912 human lung tissue sample preserved in the Berlin Museum to perform molecular clock analysis. The result pushed the divergence between measles and rinderpest back to roughly 528 before the Common Era, the 6th century BCE. That date aligns precisely with the rise of the great Iron Age cities, Babylon, Nineveh, Persepolis. The species jump occurred when human populations finally became dense enough to host a virus that needs constant transmission to survive. Cattle had been domesticated in the Fertile Crescent roughly 10,500 years ago. The viral reservoir had been waiting that entire time. Cities provided the spark. Once measles existed, it killed. The virus has an R0, or basic reproduction number, of 12 to 18 in unvaccinated populations. That means a single infected person in a community with no immunity will infect 12 to 18 others. By comparison, the original SARS-CoV-2 virus had an R0 of around 2 to 3. Measles is one of the most contagious infectious diseases known. Pre-vaccine global measles mortality is estimated at 2.6 million deaths per year, mostly children under 5. The 1875 Fiji measles epidemic, brought back from a diplomatic visit to Australia, killed roughly one-third of the entire population of the islands within 4 months, more than 40,000 people. The 1846 Faroe Islands outbreak, studied by the Danish physician Peter Panum, produced one of the foundational papers in measles immunology, but the most disturbing finding is recent. In 2019, a paper by Michael Mina and colleagues in Science demonstrated that measles infection produces what they called immune amnesia. The virus actively wipes out between 11 and 73% of the antibodies the immune system has accumulated over a lifetime, leaving survivors vulnerable to other infections for years afterward. The damage doesn't stop when the rash fades. Children who survived measles in the pre-vaccine era continued dying of unrelated infections for the next 2 to 3 years at elevated rates. Measles wasn't just killing during the acute illness. It was creating populations of immunologically defenseless survivors. Rinderpest, the cattle ancestor, was finally eradicated in 2011 through a vaccine developed by the British veterinarian Walter Plowright. It became only the second viral disease ever eliminated, after smallpox. Removing the cattle reservoir cut the morbillivirus lineage off from its only major animal source. Measles in humans has been suppressed by the MMR vaccine, developed by Maurice Hilleman at Merck and licensed in 1971. The World Health Organization estimates global vaccination prevented approximately 56 million measles deaths between 2000 and 2021. That is 6 pandemic killers. 6 viruses that killed enough people to bend the trajectory of civilizations. But the worst horror on this list isn't a pandemic at all. The next two viruses didn't kill anyone. They did something stranger, they got inside us, and they never left. Number 7. The virus that wrote itself into the mammalian brain. Bornaviruses are non-segmented negative-strand RNA viruses, family Bornaviridae. They are named after the town of Borna in Germany, the site of an 1885 outbreak that killed horses. Borna disease virus 1, abbreviated BODV1, primarily infects horses and sheep, causing fatal encephalitis. It should not, by every rule of how viruses work, be able to integrate into the DNA of the animals it infects. RNA viruses don't have the molecular machinery to do that. Retroviruses do. Bornaviruses don't. In January 2010, Keizo Tomonaga and colleagues at Osaka University published a paper in Nature that overturned that assumption. They identified what they called endogenous bornavirus-like nucleoprotein elements, or EBLN, in the human genome. These are fossilized fragments of bornavirus N gene sequences integrated into mammalian chromosomes. The integration events were dated to at least 40 million years ago, predating the divergence of Old World and New World primates. Bornaviruses had been getting into mammalian DNA for tens of millions of years. The mechanism, Tomonaga proposed, involved hijacked line 1 retrotransposon machinery. Line 1s are autonomous genetic elements that copy themselves around the genome using reverse transcription. A bornavirus that happened to be inside a cell during line 1 activity could, by accident, get its own RNA reverse transcribed and inserted into chromosomal DNA. Once inserted, it became part of the host genome. It would be passed to every descendant of that cell forever. This was the first discovery of endogenous non-retroviral RNA virus sequences in vertebrate genomes, a previously unsuspected category of viral fossils. Humans carry at least 7 EBLN loci on chromosomes 1, 2, 3, 8, and 10. One of them, EBLN1 on chromosome 10, retains an open reading frame and is actively expressed in brain tissue. Tomonaga and others have hypothesized that EBLN1 may have been co-opted by the host for some neural function, or it may not have been. The science is still active. A more contested hypothesis advanced by researchers including Hans Christian Lipkin at Columbia and the German virologist Liv Bode links bornavirus presence and EBLN expression to neuropsychiatric disorders, particularly schizophrenia and bipolar disorder. Studies have detected elevated bornavirus antibodies and RNA in some psychiatric patient populations. Replication has been inconsistent. The hypothesis is not settled. It is also not refuted. What is no longer contested is that bornaviruses can still kill humans. In 2018, the Robert Koch Institute in Germany published confirmation that BODV1 is a cause of fatal human encephalitis. The same group has identified at least 14 confirmed BODV1 human deaths in Germany between 1995 and 2018, mostly in Bavaria. For decades, these had gone unrecognized. The virus had been killing people, and no one had been looking. The endogenous bornavirus elements are passed from parent to child as part of the human genome. Every human alive carries them. EBLN sequences have also been found in the genomes of bats, rodents, primates, fish, and even Afrotherian mammals like elephants, suggesting the integration phenomenon is even older and more widespread than the original 2010 paper revealed. The Oxford evolutionary biologist Eris Katsorakis coined the term paleovirology to describe the study of these ancient viral fossils. Approximately 10% of the human genome is now suspected to derive from various ancient viral integrations, retroviral and otherwise. Most of it has not been functionally characterized. Bornaviruses are in your brain. We don't know what they're doing. The final virus on this list is the one we know exactly what it's doing, because it built you. Number 8, the virus that built your body and is still inside you. Every mammal alive owes its existence to an infection. The infection is older than primates. It is older than rodents. It is older than the lineage that gave rise to whales and the lineage that gave rise to bats. It happened in a common ancestor roughly 30 million years ago, and it is still happening every time a human is born. Human endogenous retrovirus W, or HERVW, was identified in the human genome in 1999 by Jean-Luc Blond and colleagues at the École Normale Supérieure de Lyon. Like other endogenous retroviruses, it had been written off as evolutionary debris, viral DNA that had inserted itself into a germline cell millions of years ago and been replicated mindlessly ever since, doing nothing. Then Blond's team noticed something. The envelope gene of HERVW, the gene that in a working retrovirus would code for the protein spike that allows the virus to fuse with host cell membranes, was still being expressed. Not just expressed. Expressed in one specific tissue, the placenta. The protein it produced was renamed syncytin 1. In the developing placenta, syncytin 1 causes individual trophoblast cells to fuse together into a single multinucleated cell layer called the syncytia trophoblast. That layer forms the maternal-fetal barrier. It is the cellular wall between mother and child. It controls the exchange of oxygen, nutrients, and waste. It is what makes mammalian pregnancy possible. Without syncytin, there is no placenta. Without a placenta, there is no live birth in the way mammals do it. Mammalian reproduction was built, in a literal molecular sense, by a viral envelope gene captured from an ancient retrovirus. In 2003, Thierry Heidmann's group at the Gustave Roussy Institute in Villejuif identified a second placental syncytin, syncytin 2, derived from a different endogenous retrovirus called HERV-FRD. Syncytin 2 has an additional function. It is immunosuppressive. It actively prevents the maternal immune system from recognizing the fetus as foreign tissue and rejecting it. The virus that built the placenta is also the virus that stops your mother from killing you in utero. In 2009, Anne Dupressoir's team published direct experimental proof of necessity. Mice, in which the syncytin A gene had been knocked out, died as embryos in the womb from failed placental development. The virus was not optional. Removing it killed the mammals that depended on it. Then it got stranger. Different mammalian lineages had captured different retroviral envelope genes for the same purpose. Mice used syncytin A and syncytin B, derived from a separate retroviral integration roughly 20 million years ago. Carnivores, like dogs and cats, use a third independently captured retroviral envelope. Cornelis and colleagues in a 2015 paper identified syncytins in tenrecs, opossums, and other lineages, indicating that at least 10 independent capture events occurred across mammalian evolution. Each lineage had been separately colonized by a different retrovirus. Each had evolved to depend on it. The same biological solution had been built 10 different times by 10 different infections. Approximately 8% of the human genome consists of endogenous retroviral DNA, roughly 200,000 ERV insertions. The vast majority are degraded fossil sequences. But syncytin 1 is not a fossil. It is actively expressed every time a human conceives. It is reactivated on schedule in the cells of every pregnant woman. The same gene, doing the same job that it has done in the lineage of placental mammals for 30 million years. The French biologist Cédric Fichotte at Cornell coined the term molecular domestication to describe what happened. The virus had not been defeated, it had been absorbed. The host had stolen the virus's tools and built a new biology out of them. The placenta, the Multiple Sclerosis Association studies of Antony in 2004, the cancer fusogenicity research. All of it traces back to a single retroviral integration in a primate ancestor before our species existed. You exist because of a virus. It is in every cell of your body. It is in every human alive. It is passed to your children. There is no cure because removing it means you stop existing. The virus that built you is still inside you, and it has been there longer than there have been humans for it to inhabit. A virus built your body. 2 viruses are inside your DNA. 6 viruses killed enough people to topple empires. And every virus on this list shares one starting point. They are all older than us, older than mammals, possibly older than life itself. To understand how a virus could end up building a placenta or breaking the Roman Empire, it helps to step back and ask where viruses themselves came from. The honest answer is that we don't know for certain. 3 competing hypotheses dominate modern virology. The first is the regression hypothesis, which proposes that viruses are degenerate ancestors of cells, once independent organisms that lost components over evolutionary time until they became the obligate intracellular parasites we know today. The second is the escape hypothesis, which suggests viruses are runaway genetic elements that escaped from cellular genomes and acquired the ability to replicate independently. The third, and most provocative, is the virus-first hypothesis. Researchers including Eugene Koonin and Patrick Fortere have argued that viruses predate cellular life entirely. Fortere has gone further, proposing that viruses may have invented DNA itself, with cellular organisms acquiring the molecule from their viral predecessors. Whichever origin is correct, the giant viruses discovered in the early 2000s suggest viral evolution is more complex than once believed. Mimi virus, identified in 2003 in a water tower in Bradford, England by Didier Raoult's lab, has a genome larger than some bacteria. Pandora virus genomes exceed a megabase. Pithovirus, revived from 30,000-year-old Siberian permafrost in 2014, was both the largest virus ever recovered and still infectious after 30 millennia in the ice. The boundary between virus and cell, once thought clear, is now considered blurry. The scale of the viral world is also staggering. Estimates put roughly 10 to the 31st power virus particles on Earth at any moment, outnumbering cellular organisms by an order of magnitude. Marine viruses alone kill 20% of ocean microbial biomass every day, driving the global carbon cycle. Viruses were almost certainly present in the earliest microbial ecosystems 3.5 billion years ago. The virosphere is now recognized as Earth's most diverse genetic system. Viruses are not invaders. They were here first, and we evolved in a world they had already shaped. Viruses got here before we did. They've been writing themselves into our biology and ending civilizations by two distinct mechanisms. The 8 items on this list demonstrate 2 roads by which a virus becomes permanent. The first is the biological road. Sections 7 and 8 covered viruses that integrated into the human genome and became part of who we are. HERVW built the placenta. Endogenous bornaviruses sit in our chromosomes. These viruses don't infect us in the conventional sense. They are us. The integration happened in deep time, in our ancestors, and the viral DNA has been passed through every generation since. Removing it would mean rebuilding the species. The second is the demographic road. Sections 1 through 6 covered viruses that killed enough people to bend the trajectory of civilizations. Smallpox collapsed the populations of 2 continents. HIV reshaped sexual culture, drug policy, and antiviral medicine. Yellow fever rewrote the political map of North America. Polio defined 20th century public health. The Antonine Plague broke the Roman army's manpower and accelerated the spread of Christianity. Measles became humanity's great child killer for 2.5 millennia. Most viruses do neither. They cause acute illness and pass without leaving permanent traces. A small number do one. An even smaller number do both. Measles, for example, both reshaped human population dynamics through the demographic road and may have left ancient genetic signatures in survivor populations through partial selection on immune-related genes. The boundary between the 2 roads is sharper for some viruses than others. What unites both roads is permanence. A virus that becomes part of your DNA is permanent because it cannot be removed. A virus that kills enough of your ancestors is permanent because the demographic shape of the species after it passed is not the demographic shape from before. Both routes alter the human story in ways that cannot be undone. Both qualify as rewriting human history. The 8 items on this list represent the clearest examples of each. Almost every demographic road virus on this list shares the same backstory. It crossed from an animal, and the next one will too. The pattern is now well documented. Most pandemic viruses are zoonotic, meaning they originated in non-human animal hosts and then crossed to humans. The cattle origin produced measles, rinderpest having descended from common morbillivirus ancestors. Smallpox most likely also has an animal origin, though the precise reservoir is debated. Primate origin is the case for HIV, which crossed from chimpanzees. Bird origin defines all influenza. Apandemics through reassortment events between avian, swine, and human strains. Bat origin links to SARS, MERS, Marburg, Ebola, and probably SARS-CoV-2. Rodent origin is the suspected source of hantaviruses, including the leading hypothesis for the Tudor sweating sickness. Mosquito-vectored viruses with primate reservoirs include yellow fever, dengue, and Zika. The conditions enabling zoonotic jumps are well understood. Dense human-livestock contact creates sustained opportunities for cross-species transmission. Hunting and bushmeat consumption produce direct blood exposure during butchering. Ecological disruption brings species into contact that would otherwise have remained separate. Critical community size determines whether a newly emerged pathogen can establish itself once it crosses. Wet markets, deforestation, and the bushmeat trade all amplify zoonotic risk. Of new emerging infectious diseases reported since 1940, approximately 75% are zoonotic. The World Health Organization maintains the planning category Disease X for the next unknown zoonotic pandemic with global potential. Surveillance programs like PREDICT, funded by the United States Agency for International Development, attempted to catalog unknown zoonotic viruses in wildlife reservoirs before being defunded in 2019. The pattern is not slowing. The conditions producing it are intensifying. Some of these crossings we have been able to identify. Others left graves but no fingerprints. The recovery of viral DNA from ancient remains is a recent capability. Until the late 20th century, viruses that had killed millions in the past were essentially unrecoverable. Their genomes had decayed, their tissue evidence was gone. Then, in 1997, Jeffrey Taubenberger at the Armed Forces Institute of Pathology recovered the 1918 influenza virus from preserved tissue in Alaskan permafrost burials. The pathogen of the deadliest pandemic in modern history was, for the first time, available for sequencing. The field of paleovirology had begun. The Oxford evolutionary biologist Aris Katsourakas coined the term and helped establish the discipline. Within 2 decades, paleovirology had pulled an extraordinary catalog of ancient viruses out of the bones, ice, and museum specimens. Hendloff and colleagues recovered a 14th century Yersinia pestis genome from a London plague pit in 2010. Muleman and colleagues sequenced ancient hepatitis B virus from Bronze Age Eurasian skeletons in 2018, identifying lost HBV strains that no longer exist today. The same group recovered variola DNA from 11 Viking-era skeletons in 2020, pushing confirmed smallpox in Europe back to between 600 and 1,050 of the Common Era. Dux and colleagues used a 1912 Berlin Museum lung tissue sample to date the measles divergence in 2020. Dugan and colleagues recovered variola DNA from a 17th century Lithuanian child mummy in 2016. The methods include shotgun sequencing of ancient DNA, target enrichment for known pathogen sequences, molecular clock analysis to date evolutionary divergences, and analysis of endogenous viral elements as a permanent genetic record. Frozen permafrost is a particularly important reservoir for preserving viable ancient pathogens. Pithovirus sibericum, mentioned earlier, was revived from 30,000-year-old Siberian permafrost in 2014 by a team led by Jean-Michel Claverie. It was still infectious, to amoebas fortunately, not to humans. Most ancient viruses cannot be revived but can be sequenced and reconstructed. The field has fundamentally changed estimates of viral evolution rates. Many historical pandemics still remain unconfirmed at the molecular level, including the Antonine Plague, where Roman-era ancient DNA work has so far been inconclusive. Even with all that, there are killers in the historical record we still cannot identify by name. The Plague of Athens struck the city in 430 before the Common Era, during the Peloponnesian War. The historian Thucydides survived the outbreak and wrote a detailed clinical description that has been the primary source for over 2,000 years. The pathogen has never been identified with certainty. Leading viral candidates include hemorrhagic fever, measles, and smallpox, though typhus, a bacterial infection, remains a serious alternative. The plague killed Pericles in 429 before the Common Era, contributed to Athens' defeat in the Peloponnesian War, and arguably ended the Athenian Golden Age. The sweating sickness appeared in England in 1485, the year Henry VII seized the throne at Bosworth. It killed within hours of the first symptoms, struck in 5 distinct waves over the next 66 years, and vanished after 1551. It has never been confirmed. The leading hypothesis is a hantavirus. Other proposals include enterovirus, anthrax, and ergot poisoning. In 1502, the disease killed Prince Arthur Tudor, the elder brother of the future Henry VIII. Arthur's death made Henry king. The English Reformation, the dissolution of the monasteries, the Tudor succession crisis, all of it traces back to a virus we still cannot name. Encephalitis. Lethargica struck the world from 1915 to 1926. A pandemic of mysterious encephalitis left an estimated 1 million people in catatonic frozen states for decades. The pathogen has never been confirmed. Viral cause remains the leading hypothesis, though some research has pointed to a streptococcal autoimmune mechanism. Oliver Sacks documented the survivors in his book Awakenings, later adapted into the film of the same name. Outbreaks may be returning. The Cocalistli of Mexico, 16 waves between 1545 and 1576, killed approximately 80% of the surviving Aztec population after the initial smallpox catastrophe. Recent ancient DNA work by Vaugigny and colleagues in 2018 identified Salmonella enterica parodifi C as the likely culprit, bacterial, not viral. The viral hemorrhagic fever hypothesis is now considered less likely. The case demonstrates how identification can dramatically reframe what we thought we knew. Each of these is a candidate for a future version of this list. The science is still catching up, unnamed killers in the historical record, and somewhere right now the killer that future historians will name first is already moving. The conditions that produced every virus on this list are intensifying. SARS-CoV-2 emerged in late 2019 and has caused approximately 7 million confirmed deaths globally as of recent reporting, with the true toll likely higher. Its long-term historical impact is still being written. Economic, political, and social shifts visible in the wake of the pandemic will take decades to fully understand. Whether SARS-CoV-2 will eventually be added to lists like this one is not yet clear. The vote is being cast now. H5N1, avian influenza, has been watched closely as a potential next pandemic since the late 1990s. Recent spillover into United States dairy cattle in 2024 has raised serious concerns about mammalian adaptation, a critical step for sustained human-to-human transmission. Marburg outbreaks were recorded in Equatorial Guinea and Tanzania in 2023. Ebola continues to emerge from West African and Central African forest reservoirs. The mRNA vaccine platform developed for COVID-19 has reshaped how new viruses can be countered, but pandemic preparedness funding and political will fluctuates dramatically between crises. The conditions producing pandemic viruses are intensifying. Climate change is shifting vector ranges. Yellow fever and dengue mosquitoes are moving poleward. Deforestation is pushing new species into contact with humans, increasing zoonotic spillover risk. Factory farming creates evolutionary pressure cookers in which influenza strains can reassort and gain new properties. Urbanization concentrates susceptible hosts. Global travel collapses the geographic fire breaks that once contained outbreaks within continents. Genomic surveillance systems can now catch new pathogens within weeks. The technology has improved enormously. The institutional and political response capacity has not kept pace. The emergence of the next history rewriting virus is essentially guaranteed. Only the timing and identity remain unknown. 8 viruses on this list. 6 of them killed empires, 2 of them built you. And the 9th one, the one that will join this list in a century, is already out there. Humanity is not the protagonist of a story occasionally interrupted by viruses. Viruses are co-authors of the human story and have been since before our species existed. They were on Earth before a complex life evolved. They wrote themselves into the genomes of our ancestors. They built the placenta that you were born through. They sit in your brain, in your chromosomes, in every cell of your body. They built the religious history of the Roman Empire, the political map of North America, the demographic shape of the Americas, the medical infrastructure of the 20th century, and the cultural movements of the 20th and 21st. Smallpox killed roughly 400 million people in the 20th century alone before eradication. HIV has killed 40 million and counting. Yellow fever defeated Napoleon's Caribbean army and forced the Louisiana Purchase. Polio shaped 20th century public health and Roosevelt's presidency. The Antonine Plague accelerated the spread of Christianity. Measles became humanity's great child killer for 2.5 millennia. Endogenous bornavirus elements are sitting in your chromosomes right now. We don't know what they're doing. Syncytin built the membrane that kept you alive in the womb. You cannot remove it because removing it means you stop existing. Every breath you take, every heartbeat, every cell division is happening in a body partly built by ancient viruses in a world where their descendants are still writing chapters. The next one is incubating right now in some animal you have never seen, in some region you have never visited. It will reach you eventually. You won't know it until later. You are what the viruses made you, and they are not done.